Hello everyone, my name is Wang, and my advisor is Professor Zhang Xunshuo. And today I'll be presenting the final report for my advanced laboratory training course. And this semester we had the opportunity to rotate through different laboratories, and I chose to focus on the laboratory of Assistant Professor Xie Kanyan. The topic I'll be discussing today is foundation of protein drug, recombinant protein and bacterial transformation. Throughout this presentation, I'll explore not just the technical procedures, but also the underlying principles and their broader implication for drug development. Today, I'll first introduce why protein drugs is important and why recombinant production is required. Then I'll briefly describe the experimental design, including plasmid components and the chemical transformation workflow. Next, I'll present the experimental result and discuss their implication for protein drug development. Finally, I'll share my reflection and take-home messages. First, introduction. Before discussing the technical details, it's essential to understand why recombinant proteins have become so important in pharmaceutical research and drug development. According to recent data, biologic drugs make up around 30% of all new FDA approvals each year. This demonstrates how central protein drugs have become a modern medicine. And many essential medicines we use in clinical practice today are proteins. For example, insulin for diabetes management, and growth hormone for treating growth disorder, and interferons for immune system modulation, and monoclonal antibodies for treating cancer and autoimmune diseases. These are all protein-based therapeutics. And what makes protein drugs so valuable? Compared to traditional small molecular drugs, protein drugs offer several distinct advantages. First, they exhibit high specificity. Protein can be designed to interact very precisely with specific biological target, like a key fitting into a lock. Second, they show strong biological activity, meaning they can achieve therapeutic effects at relatively lower concentration. Third, because of their high specificity, protein drugs tend to have fewer off-target effects. They are less likely to interact with unintended molecular targets, which often translates to better safety profiles. These advantages make protein drugs a powerful and increasingly indispensable class for modern medicines. However, producing protein drugs directly from nature's sources is often not feasible, which leads to the need for recombinant production. And although proteins can be isolated from natural sources, this strategy is generally not suitable for protein drug development. First, many bioactive proteins exist at very low and highly variable levels in natural organisms, which make large-scale production difficult. And second, pharmaceutical manufacturing requires highly scalability and strict batch-to-batch consistency, which are difficult to achieve when relying on natural sources. In addition, protein isolated from biological tissue may carry contaminants such as host proteins or endotoxin, raising safety concerns. Finally, nature isolation does not allow molecular design or sequence modification, which is often required to optimize therapeutic efficacy. To overcome these limitations, recombinant protein production is widely used in modern pharmaceutical development. In this approach, the gene encoding the target protein is first inserted into an expression vector, typically a plasma. The engineering plasmid is then introduced into a suitable host cell, such as E. coli, which serve as a controllable biological factory for scalable protein expression. This system allows precise control over protein sequence, expression level, and production scale, making it highly suitable for therapeutic applications. In the next part, I'll briefly introduce the plasmid design and the bacterial transformation process used in this experiment. Materials and methods. This slide shows the basic components of an expression plasmid used for recombinant protein production. And first, the origin of replication determines how the plasmid is maintained and copied inside the host cell, which directly affects plasmid copy number and protein yield. Second, the promoter controls when and how strongly the target gene is expressed. In protein drug development, inducible promoters are commonly used to balance cell growth and protein production. Downstream of the promoter, the ribosome binding sites ensure efficient translation initiation. The gene of interest anchors the therapeutic protein or protein precursor. And finally, the selectable marker, such as an antibiotic-resistant gene, allow us to selectively grow only the bacteria that have successfully taken up the plasmid. After designing the expression plasmid, the next critical step is introducing this plasmid into the host cell through bacterial transformation. And chemical transformation is a widely used method to introduce plasmid DNA into bacterial cells. At low temperature, plasmid DNA associates with the bacterial cell surface under chemical conditions that reduce electrostatic repulsion. The key event in chemical transformation is heat shock. Induced membrane permeability, which allows plasmid DNA to enter the cytoplasm. After transformation, cells are allowed to recover in non-selected medium, during which antibiotic-resistant genes are expressed. Only cells that have successfully taken up the plasmid survive under antibiotic selection and can subsequently express the recombinant protein. And this slide illustrates the chemical transformation procedure. First, plasmid DNA mixed with the component cell and incubate on ice allow DNA to interact with cell membrane. And heat shot at 42 Celsius temporarily increase the membrane permeability. And finally, the cells are recovered in LB medium to obtain transformation cell. And you may wonder how negatively charged plasmid DNA can enter bacterial cells that also carry a negative surface charge. Under normal conditions, this electronic static repulsion prevents DNA from entering the cell. To overcome this barrier, bacteria are first prepared as chemically component cells by treating with calcium chloride. And the calcium ion neutralizes the negative charge on both the plasmid DNA and the cell surface, allowing the DNA to associate with the membrane. A brief heat shot is then applied, which temporarily increases the membrane permeability and enables the plasmid DNA to enter the cell. Next, results. And this slide shows the experimental result of the bacterial transformation. On the left is my transformation plate, where visible colonies indicate successful uptake of the plasmid and expression of antibiotic resistance. On the right is a comparison among different groups in the class. While some plates show abundant colonies, others show few or no colonies at all. These differences suggest the variability in transformation efficiency under similar experimental conditions. Discussion. Transformation failure can occur at multiple stages of the process. If plasmid DNA does not efficiently associate with the bacterial cell surface, uptake will not occur. In addition, heat-shot conditions that are too mild may fail to induce membrane permeability, while excessive heat shot can reduce cell viability. Finally, insufficient recovery time may prevent the expression of antibiotic-resistant genes, leading to cell death on selected plates. Therefore, successful transformation requires a balance between membrane permeability and cell survival. And colony number is commonly used as an indicator of transformation efficiency, reflecting how many cells successfully took up the plasmid and survived antibiotic selection. And the colony size, on the other hand, is primarily influenced by two major factors. The first is cellular growth rate, which can be affected by plasmid copy number and the metabolic burden associated with recombinant protein expression. The second factor is local nutrient availability. When colony density is low, individual colonies experience less competition and may grow larger, even if their intrinsic growth rate is not particularly high. And despite this limitation, these experiments provide important insight into protein drug development. Early-stage processes, such as bacterial transformation, directly affect downstream protein yield by determining the number of productive colons. In addition, recombinant protein expression in-process metabolic stress on host cell, which can influence growth and overall productivity. Therefore, protein drug development requires careful optimization on both genetic design and biological process. Reflection. As far as I am concerned, this course expanded my understanding beyond nature product isolation to the foundations of protein drug development. As a student trained mainly in nature product chemistry, I usually focus on the compound isolation and structure elucidation. However, through these experiments and the laboratory rotations, I experiment how biological variability and process control play critical roles in recombinant protein production. Overall, this course not only introduced me to new experimental techniques and instruments, but also helped me see how genetic engineering, biological system, and pharmaceutical development are closely connected. Although the early morning schedule was challenging, this course turned out to be one of the most rewarding and engaging laboratory experiments for me. Thank you for your attention.